Ladies and gentlemen, welcome and thank you for joining today's call, New World Screwworm. Please note that all participant lines will be muted throughout the call. We will have Q&A at the end of the call, and I will provide instructions on how to ask a question at that time. If you require technical assistance, please send a private chat to the event producer via the chat panel located on the lower right-hand side of your screen. With that, I'll turn the call over to Liz Fernandez. Liz, please go ahead.
Welcome, everyone. I'm Liz Fernandez with the Professional Development Staff, and I'd also like to welcome you to today's webinar. We have two speakers today. Our first speaker will be John Welsch. John currently serves as the Liaison for Action Programs for International Services of APHIS USDA. John is a liaison between APHIS IF, International Services, and other U.S. and international governments, universities, and non-government agencies. He began working on Screwworm in Mexico as a postdoc in February 1984 and was hired by USDA ARS Screwworm Research Unit as a Category 1 research scientist in December of '84. John conducted research on the ecology of screwworm in the tropics for 18 years and served as research leader of the USDA ARS Screwworm Research Unit until 2001. He also served as technical subdirector from 2001 to 2002 and U.S. director of the Mexico-United States Commission for the Eradication of Screwworm from 2002 to 2005. He served as a technical director from 2010 to 2015 for Panama United States Commission for the Eradication and Prevention of Screwworm and continues as technical advisor and serves as the OIE expert at the OIE Reference Laboratory for New World Screwworm from 2011 to 2018. John has lived and worked in the field on screwworm in Belize, Honduras, Nicaragua, Colombia, Jamaica, Aruba, Cuba, the United States, Uruguay, and Argentina. During the 2016 and 2017 outbreak of screwworm in Florida, he served as a subject matter expert and led field operations in surveillance and eradication of screwworm. At the request of the government of Uruguay, he assisted in the development and execution of a field study to examine the efficacy of the Uruguay River as a barrier to screwworm fly dispersal between Uruguay and Argentina. Our second speaker is Pamela Phillips. Pamela came to work for ARS in 1994 with a strong background in remote sensing and GIS technologies with expertise in habitat mapping in tropical environments for the World Wildlife Fund, the Nature Conservancy, and Tropical Research and Development. Her first position in ARS was in Panama City, Panama, studying the field ecology of native screwworm populations and resulted in a better understanding of seasonal and spatial distribution of screwworm and their habitats in tropical areas. These results have been applied to the APHIS Screwworm Program to direct field operations, plan patterns, rates of both aerial and ground dispersal, release of sterile flies, direct fly trapping activities, and more recently identify areas for a collection of native flies for genetic study and new strain development. In 2012, she was assigned to work in Panama again, this time at the Pecora sterile fly production facility. With the supervision of Steve Sirota and knowledge of other ARS colleagues, she learned about rearing screwworm flies, the production of screwworm flies at the facility, and along with Steve, facilitated in the on-site management of transgenic screwworm studies. She had a terrific career with ARS, but now has taken a position with APHIS IF Action Programs to serve as the technical U.S. director at COPEG in Panama. She oversees the operations of all plant and field sections to meet the mission, objectives, and goals of the APHIS IF Screwworm Action Program. And with that, I will turn the webinar over to John Welsch.
Thank you, Elizabeth. Good morning. I've been asked to talk a little bit about the biology of the New World Screwworm and the history, and including the surveillance and how that has changed throughout time, and the general eradication strategies, which the main one is the sterile insect technique. So thank you all for joining the program, and here we go. Screwworm is endemic in the Western Hemisphere, and it's found between 41 degrees 58 minutes north latitude and 38 degrees 03 minutes south latitude, except from the areas where it has been eradicated. When the Europeans first started coming to the New World in the middle of the 16th century, they came into acquaintance with this obligate parasite, and they had not seen anything about it previously, and so it made its way into literature at that time. One of the things that the Spanish conquistadors did was oftentimes if they had an unruly prisoner that they had taken, they would brand the prisoner on the cheek with the letter T, and they reported that oftentimes these maggots or worms or gusanos would miraculously appear coming out of these wounds, and eventually the prisoner would die from this type of infestation. So that was back in the middle of the 1550s, and then if we jump ahead about 300 years, the French government became aware of a problem with screwworm, and they didn't know what it was to begin with. They had a penal colony in French Guiana, and it was called Ile de Diablo, or Devil's Island, and something was eating the prisoners alive. And so they sent a physician and scientist, Dr. Charles Cockerell, to the penal colony to try to figure out what was going on. And so Dr. Cockerell studied the situation and found that it was caused by a fly, and he named it originally Lucilia hominivorax, which Lucilia means shiny and hominivorax means man-eater. However, due to the taxonomy of the scientific taxonomy, the name eventually became Cochleomya hominivorax, and Cockerell is the first person to describe it, and is also known as the primary or New World screwworm. He described how he found the wounds, and they were infested with hundreds of these maggots, and he pulled them out, and he saw that they had these mouth hooks, which you can see in the bottom two pictures, the dark, heavily sclerotized mouth hooks, and the maggots use this to scrape away at the flesh, and you frequently can have wounds that contain hundreds to thousands of these animals, and when they reach their maturity, they're about 3/4 of an inch long, so they do considerable damage. And here's a photograph of a female at the top laying an egg mass. That's a bright white mass behind her, and then a male at the bottom, and you can see how they're shiny, and thus he gave them the names Lucilia. The females, if you notice on the top left, their eyes are separated, whereas the males, they're almost touching. And the males visually locate the females as they fly by and then tackle them in the air to mate with them. Dr. Cockerell also was the first person to kind of determine the life cycle, and then it was later detailed by other scientists. The female fly can lay an egg mass of 200 to 300 eggs. These eggs hatch after about 10 to 12 hours into almost microscopic sized larvae, and the larvae invade the wound or any other bodily orifice wherever the egg mass is attached. And they feed in the wound for 5 to 7 days, growing again from pretty much microscopic to 3/4 of an inch long each. When a wound is infested with larvae, it gives off a distinct odor, which is then more attractive to other gravid females or females needing to lay eggs, and so you get multiple infestations. After the 7 days, 5 to 7 days, the larvae crawl out of the wound, drop to the ground wherever they are, and they try to burrow in, and they form the pupae. And the pupal stage, depending on local weather conditions and such, lasts anywhere from about 5 days to on up if it was wintertime in a cold area. But generally during the summertime, 5 to 7 days. And then the flies, they emerge, and after about 3 days, they're ready to mate, and then they mate, and the female begins to oviposit. During her lifetime, the female can oviposit. We've documented up to 10 times in the field, again laying 200 to 300 eggs each time. So that's back in 1858 when he discovered the fly. Well, it wasn't until 1933 that Emery Cushing, the gentleman there in uniform, studying this insect he had taken some samples with him to Oxford in England, and he was able to determine that there were two different species in what he had collected. And the one, Cochlea hominivorax, the primary screwworm, was the one that actually did the damage to the animal, and that's being called primary. And then the secondary screwworm was just another insect that had caused the myiasis and was actually feeding on necrotic tissue, and that was available. Well, this was a major breakthrough because up until that time, nobody knew the difference. One thing I also like to mention right now is, we said that Cockrell identified the fly in 1858. Well, there's a report from an Army lieutenant, head of a cavalry detachment in the United States in the mid-plains, the Great Plains area in 1860, and he described that they had a problem with some sort of worms infesting a lot of their horses, and it's often thought that might be the first report of screwworm in the United States. But of course, at that time, nobody knew the difference between the flies or anything species. So, previous to Cushing, they used traps to try to control screwworm. There, in front of the car on the left, there's a bishop trap, and it's baited with rotting liver and hung out there. It has an inverted cone. And then the mountain next to the guy, those are all flies. And this is what they did to control screwworms in the 1930s, and it didn't work. It's just pretty amazing. Traps have changed throughout time. Big change happened in the 1970s. Alberto Brose developed what was called the wind-oriented trap, which was a plastic bucket with an inverted cone, and they used a chemical formulation, a bait called swarm lure for screwworm lure. And this trap here was one that was in Costa Rica that we placed out in the field, and it used the fourth generation of the formula, which was developed by Mackley and Brown in 1984, and so it was called Swarm Lure 4. And Swarm Lure 4 is still the chemical attractant that we do use in wind-oriented traps to collect the flies. Screwworms have been collected from sea level up to 7,800 feet in altitude. They're generally active between 64 degrees and 91 degrees Fahrenheit. Most activity is between 78 to 91 degrees, and peak activity that we found in Costa Rica was around 85 to 86 degrees. Generally, there's no activity below 15 degrees Celsius or 59 Fahrenheit, but there are reports of oviposition in the field at 61 degrees Fahrenheit. Generally, the mean annual temperature, if it's lower than 66.9 degrees, you're not going to have a problem with screwworm. Also, during times of the years where the mean monthly temperature is lower than 48.9, it won't be a problem. The absolute minimum temperature that they could survive is 20 degrees Fahrenheit. Now, populations generally are higher in areas with annual rainfall greater than 59 inches, and generally the lower limit is considered to be approximately 6 inches of rainfall for populations to occur. Generally, screwworm prefers wooded areas over pastures or fields, and especially during the dry season, They're confined in the riparian habitats along rivers and streams, and this is also where the hosts and both the wildlife and the domestic animals, livestock and other animals, go up into the cool shade and to the water to rest, and of course the flies are there waiting for them. Screwworm flies are found clustered. They're not evenly distributed within the habitats. You'll have activity at one site in one day, and then it might be quiet the next, and then they'll move back in the next day. They're moving about. They're only active during the daylight, and their activity slows down on cloudy days. When they do not like windy situations, and again, this is also why I believe they prefer the wooded areas and riparian habitats, their activity ceases. They hang on to the vegetation with winds exceeding 15 miles per hour. Generally, females only mate once in nature, and that is way to our advantage and has been a key to the success of the program. Under the best of conditions, screwworm is present at only 100 to a few hundred per square mile. Now, to put that in perspective, there have been reports and estimates of mosquitoes, the density being 70 million per square mile during a swarming behavior. And so screwworm is relatively rare compared to other species of insects. And this is kind of unique, and also this is to our benefit. Now, although it is rare, again, remember the mouth hooks, and remember the larvae all rasping away at the flesh of the animals. And I'm going to show a few slides here just to remind us what the damage looks like. Some of you have seen this before. Others have not seen an infested animal. And so here's this photograph has been in use since the 1970s. This does show a wound in the area of the eye that is a multiple infestation. So it was given off the odor, and more screwworm females arrived and laid more egg masses, and they hatched, and they start their cycle, and this continually goes on until either the wound is cured or else the animal dies, either directly from the wound or as susceptibility to predators or accidents or secondary bacterial infestation. Screwworm is a slow death sentence and a very horrible one. This is a dog in Jamaica, and I counted over 1,000 larvae, third instar, very mature larvae in the wound. Again, multiple infestations. This is a deer from the outbreak in the Florida Keys in 2016, 2017. And I want you to note that that deer is blind. Its eyes are eaten out. I'm sure it's deaf. And that deer was euthanized moments before this photograph was taken. So this animal was, I can't imagine the suffering, but it was unable to drink or eat or anything. And this, again, is reasons why we need to take this seriously and in keeping this out of the United States, but also trying to eliminate it from other areas to help other people, and also to reduce the risk of reintroduction. The next page I'm going to just put on is, again, the scientific name is Hominivorax maneater. These are human infestations. As far as I know, all these humans were cured about this. On the human side, generally it is the people that cannot take care of themselves for one reason or the other that are infested. They're mentally incapacitated, or they're very young or very old. They have diabetes or something. There's something, but also it can be in just a very healthy person. So anyway, it runs a gamut. So anyway, here's there's an oral infestation, ocular, person had head lice and got screwworm, and then person cut his arm in construction. They bandaged it up and it got screwworm. So It does have an impact. Human cases are typically underreported because people are embarrassed about those. But it's nothing they should be embarrassed about. It's something that we should be aware of to, again, increase the support of getting rid of this animal. The estimated annual benefits were calculated in 2005 by the FAO, and I converted the 2005 figures into taking into account inflation. And so in brown are the figures that they came up with, and in blue are the positive, that's the annual benefits, and in red are the losses. So the estimated annual benefits to producer, $1.7 million in today's dollars. And then that's to the producers alone. Then it gets multiplied as you go into the consumers and such. And so it is very positive. Estimated annual losses to South America and the Caribbean, $4.5 billion a year. So, I mean, it's just incredible. In addition to the suffering, it causes this economic impact, which raises the price of meat and food and the use of insecticide and misuse of insecticide. In 2005, it was estimated the total cost of the program, and I just, again, converted it. And so the total cost from inception to right now is approximately $1.7 billion. So it's very cost effective. Okay, well, after the Spanish entered entered the New World and populations increased and that screwworm was taken into the United States. This is the normal summer spread during before 1933. It was limited during the winter times and due to the severity of the winter or the mildness of the winter. In 1933, there was drought conditions in Texas, and a lot of cattle were shipped to Georgia and Florida, and unfortunately, some of them were infested with screwworm and took it along with them, and then the eastern part of the United States became infested with screwworm. Up until that time, evidently, the Mississippi River served as a barrier to the natural limitation of the populations. Screwworm will cross water, but screwworm does not normally cross water. And so this was a barrier before then. Well, in the 1930s, the U.S. government got interested in the situation, and they started doing work developed by the Agricultural Research Service. Melvin and Bushland worked together in developing mass production techniques. They found a way to rear the maggots off a living host. There in the top picture, the person on the far right, closest to us, is Nippling. The next person is Arthur Lindquist, and it's Raymond C. Bushland. And I believe Melvin is bending over there with his head facing us. I'm not sure because you can't see his face, and also I don't know what Melvin looks like. I've never met that gentleman. So anyway, but in 1936, Bushland and Nippling led the team to really get screwworm moving. One thing I want to also say is that Edward Nippling got his, I'm calling you from College Station, Texas, and Edward Nippling got his bachelor's degree in entomology here in 1930, and so 6 years later he's leading the team to address the screwworm situation. And it was Edward Nippling who came up with the idea of the sterile insect technique. Again, all this work was team efforts, but he was a leader, and they were able to produce large numbers of insects, and they thought, okay, well, if we could do this and sterilize them and release them over areas where the endemic populations were, they should be able to do it. And this is where, again, it was in the favor of the program that screwworm occurs in very low numbers compared to other insect and other insect pests, and also that they only mate once during their lifetime. And they also used, they pioneered the use of irradiation to sterilize the insects. Nippling was able to calculate that if you produced and released 10 sterile males for every one wild male, that it would cause the population to begin its decline and then rapidly crash and then go into extinction. And this has held true since the inception of the program, and we've estimated populations during the progress of the eradication program, and the 10-to-1 ratio still holds. And I just find that, you know, fantastic and amazing that he figured that out. So they were working on it in the 30s, and then World War II resulted in everything being put on hold, and nippling and bushing were reassigned by USDA to work on mosquito control to support the U.S. military operations. Well, after the war, they began studies on field testings of release of sterile insects, and they tried the test in Sanibel Island, right off the coast, Gulf Coast of Florida, and they were able to achieve about 80% sterility, but they were not able to drive the population into extinction. The island is very close to the mainland, and it's got a large bridge, and the flies were crossing over, or infested deer were crossing, we don't know. However, the governor of Curacao, off the coast of Venezuela, he heard about that, and they had screwworm in Curacao, and he contacted Nippling and said, hey, would you like to come down here and try your test here, because they're further off the coast, and he felt that the conditions there might be favorable, and the governor offered support, and USDA supported that. And so they did a full test on trying to eradicate screwworm, and they were able to do that successfully. And so this was really the inception, the ball started rolling. Jack Campbell heard about the success. He was the chief veterinary officer of the state of Florida, and he and the Florida Cattlemen and Livestock Producers Associations got together and lobbied U.S. government for a program. And so they began releasing sterile flies, producing sterile flies initially in Bithlo, Florida, but then they built a plant in Sebring, Florida. And during the time period of 1957 through 1959, screwworm was eradicated from the eastern portion up to the Mississippi River again. And have to show you the photograph again, talking a little bit about other parts of the techniques. Originally, the pupae were boxed into these little cardboard cartons, and the flies emerged while they were in the carton, and the pilot would fly, and a disperser would fly with them, and they would just throw the boxes out the window. I also like to say that the person on the right, the person in the middle with the light pants and the dark shirt, his name is Hugh Graham, and he's actually the guy that hired me many years ago. And this photograph was taken in 1958 in Florida. Here's a photograph of a back end of a plane. This is later during the program, and it shows in upper left, you can see a box in the chute, and it comes out of the conveyor belt, and you see the wind, and there's a cutter there, and the wind tears it apart, and then the flies are released in that manner. That was used, the box flies were used and can still be used if necessary, but we switched over to using chilled flies, where the flies are emerged on the ground and chilled down and put in to the planes cold and torpid, and then they're passed out of the planes as the adults. And so it eliminates costs of boxes and a lot of other aspects. Well, as did the Florida Cattlemen's Association, that after they heard the success of Curacao, well, the Texas and Southwest livestock producers saw the success of the program in the eastern United States, and they lobbied, and a person really instrumental in this was Charles Scruggs, and he wrote the book The Peaceful Atom and the Deadly Fly. If you get your hands on it, it's a very interesting read because it describes the history of the program to that date. Charles, his fly logo there on the book was taken by the program to use. It was stylized, and it's used as an atomic fly. Essentially, 1962 was a big year. Funding was achieved. Documents were signed. The plant, another big plant, was built in Mission, Texas, and it was dedicated in 1962. So from 1962 to 66, screwworm is being eradicated from the Southwest. In 1966, the U.S. was declared free. And then the program entered into what was called the barrier phase, where they released to try to keep the flies coming out from Mexico. If you look down at the bottom, you'll see a bar, a vertical bar going across. That would become the second barrier during the program as it progressed further to the south. In 1972, there was a big explosion of screwworms in the U.S., and the colored area, you see the stippled area, that was essentially the barrier zone. So it was massive in size, took a lot of time and money to do, to maintain it. So a treaty was signed with Mexico, forming the first international commission between the United States and a foreign country to eradicate screwworms, in addition to Curacao. And in 72, they started construction on the plant in Mexico, and they used the plant in Mexico to eradicate all of Central America. Mexico was eradicated by 91, and then the Panamanian Commission was formed in 1994. Here's a photograph of the plant in Mexico. It was in operation from 73 to 2012. And I just included this to give you an idea of the magnitude. That plant was able to produce 500 million flies per week. When I was the director there, we were producing about 200 to 250 million flies per week. And this is how much diet materials we used to rear the flies and to produce the sterile flies. And so this is a massive operation, 24/7, 365 days a year. The program continued to pass down the south.
Your connection is now in listen-only mode.
Reached the barrier zone, the vertical line that I showed you in the other photo. The cattle ranchers in the southern and eastern part of Mexico, Chiapas and the Yucatan Peninsula, they demanded, well, hey, we don't want screwworm. And so they increased the program into that area. And as a result, there were some visionaries, and they said, you know, it'd be a lot less expensive and further away from the U.S. if we pushed it all the way down to Panama and maintained the barrier zone there. And one person I'd like to acknowledge is Dr. John Wyeth, and he was instrumental. He came up with all the agreements with every one of the Central American countries. I think he was as much of a visionary as Knipling and Bushland, and he was able to get the job done politically and logistically, so he pushed it down to the barrier zone in Panama, and there's the plant in Panama. The plant in Panama can produce approximately 100 million flies per week, sterile flies per week. We have a dispersal center in Panama. Again, we don't throw flies out the window anymore in boxes. We use the chill fly technique there in the upper right photo. They're cold. They're about 10 degrees Fahrenheit. No, excuse me, 10 degrees centigrade. Put onto the plane, and it uses an auger system. to release them. Previously, all the flight lines were drawn by hand, as seen on the left. On the right, this is in 2003, and the pilots would fly with a map on their lap and with lines drawn and notes on there. And nowadays, it's all done by computer. You can see a plane icon below the red dot up in the top part, and then flight lines, and everything is programmed, and it makes it a lot more efficient, and we keep track and make sure all the areas are covered as needed. Other forms of surveillance changing through time. In the 90s, I got the idea that a dog could be trained to detect screwworm-infested animals. Again, they have that unique smell, and so I was assigned the task, and I was able to do it. Also in the lower right, because of the dog, I noted that, and I tried it, he could smell. There's a difference in the smell between an infested wound and the screwworm pupae. And so with a different term, I used one-term search for him to locate an infested animal. If there's an infested animal in the vehicle or the corral, on the upper left you can see him standing up, and on the lower left standing up. That indicates that there's an infested animal in there. And then on the right, upper right, he ignores the vehicles because there's no screwworm there. And then the lower right, the upper right, lower right is Castador looking for pupae and buried in the soil. Also, the traps continue to change with time. Renee Garcia and I worked together in developing what we now use, and it's called a vertical sticky trap, and it catches more flies than Brose's Watt trap. It still uses the chemical attractant swarm lure. It's one of our tools, but again, still our most effective tools. You go back to the 1930s, and we just, Frank Parker was a scientist that worked with us in in Costa Rica, and he took the liver out of the trap and we set it on the ground, and it's labor intensive, but this is the way we collect more flies than anything. Another type of trap or way to collect flies was the development of artificial wound. This was done by Federico Pudavine and Isidoro Ruiz in 1995. Essentially, it's a hot water bath which maintains bovine citrated buffered blood that has been inoculated with 12 species of bacteria that were isolated from screwworm-infested wounds. And actually, gravid females would come in and oviposit on the trap, and you could collect them and you could trap them by putting sticky stuff. However, logistically speaking, it was a bear, and it's just not feasible. The middle photograph on the bottom, Castador, the detector dog, he would respond to the artificial wound just as if it was an infested animal. So we thought that was pretty cool. This is our current state. We do have screwworm still present in the Caribbean, Cuba, Dominican Republic, Jamaica, and then down the lower right portion, Trinidad and Tobago off of the coast of Venezuela. Then, of course, we have all of South America, and this serves as potential for bringing screwworm into the country. Even though the Caribbean is closer, screwworm was introduced into Libya in the late 1980s, and the source of the infestation or the introduction was found to be the country of Uruguay down in the southern part of South America. So as long as screwworm is present, it's a potential problem. On the left, you can see the presence of screwworm before the eradication program, and then on the right, the current distribution. This is recognized by the FAO and the OIE and the IEA as a transboundary disease, and it's listed number 6 on the FAO's list of transboundary diseases and risks and potentials. Challenges, out of sight, out of mind. We're a victim of our own success. However, we have to maintain the vigilance. Outbreaks in the U.S. are unlikely to occur, but when they occur, the consequences are high. Right now, we have no backup plant. We only have the plant in Pecora. We need to, the countries where we've eradicated screwworm from, they are not really maintaining surveillance as we would like because, again, out of sight, out of mind, they don't have screwworm anymore. Why do we need to look for it? We continually try to engage them, and we do, and so we are in that situation. We need to maintain and develop expertise, and this is an area that we really need to work on. And, of course, obviously we need funding, continued funding, and not continually reducing our budgets. All in all, this statement, if you read this by Nippling, I think if you look at this, we could look at the statement today and say, well, we need to look at South America like this. We can either continue to fight introductions of screwworm into Florida, into Libya, into other places, or we could take the measures to eradicate screwworm from where it still exists and then completely eliminate it. I would like to acknowledge everybody who's contributed to the eradication of screwworm, not only the government workers, the scientists, but all the people in all the countries. This has been a huge team effort, and everybody who has to deal with screwworm knows what it's like, and they generally do everything they can. And again, I would like to thank you, and I will now turn it over to Pamela. Thank you again.
Good morning, everyone.
Pamela, go ahead and hit escape,
yeah.
And then you can.
Yeah, I'm hitting escape, but I still have Dawn's presentation up.
Yes, you need to click on the tab that has your name PLP on the upper part of the content window.
Oh, here we go. I go to return. Okay, good. Okay.
And then just go to your first slide.
Right. Okay. All right, good morning, everyone, from Pecora, Panama. Nice day here, a little bit cloudy, rainy season starting. I'm going to continue this presentation by talking a bit about the ecology of screwworms and going from field observations in the 30s to what we use now, which is satellite imagery. Ecological studies began really in kind of 1933 to 1950, mostly focused in Texas and Arizona. These were done by the USDA Bureau of Entomology and Plant Quarantine, later became Agricultural Research Service. The habitats that they were working in were semi-arid brush. From 1936 to 1940, they did yearly surveys, and they had essentially a mean of 1,500 cases of screwworm a year, and this was just in 2 counties in Texas. Now, at that time, the production was mostly sheep and goats. So there was not that much cattle there, but it was a huge number of cases that they had, and I think it's because of just the husbandry of typical husbandry of sheep and goats is not tended quite as much as cattle is. At that time, the preferred habitat was thought to be in open pastures. That's where they had the cases, so that's where they looked for things. And it was also recognized that the distribution was spatially aggregated across the landscape, as John mentioned as well. Now, the scientists in Menard County were Parrish, Cushing, and Lockheed, and Uvalde County, Lindquist, Parman, and Leonier. Probably didn't say that correctly, but so they all had really big contributions that they made to the program, understanding the ecology. One of the biggest things that they figured out was the importance of temperature and precipitation and the ecology of screwworm. And this was joint work. It was really understanding the roles of temperature and humidity play. They would release flies at 76 degrees Fahrenheit, and they were active. When they would release flies at 65 degrees Fahrenheit, they were not active. The overwintering also in Texas, it was considered a 5-month period from November to March. It had a limiting factor of the temperature. If it went below 53 degrees in that period of 5 months, they would have a different result, and it would kill some of the teepee that were probably overwintering in the ground. As John mentioned, the lowest temperature for oviposition was 61 degrees Fahrenheit. They also found if there was any kind of abrupt or sudden change in temperature, that it would kill a high percentage of the adult flies. They also found that in drier years, there was less abundance of screwworm. So during that 5-month period of overwintering, if they had very little rainfall, that would also influence the number of cases that they would have in the area. Barrett, in 1937, did a study looking at the natural dispersion of Cochlea maria. At that time, they called it Americana, the species Americana, although we now know it's omnivorous. I don't have a pointer here, but the pink area is Uvalde County. The yellow area is Menard County. That's where most of the research was done. Leonier discovered that, or determined that the area of overwintering is that line and kind of in between there, the brown line you see on this map of Texas. That's the Balcones Escarpment. That north of the Balcones Escarpment, there was no overwintering. This study was looking at positive cases as they spread through the winter. So the first case in 1936 showed up in Uvalde County in March, the 5th of March. It was the first fly present. If you looked at the scale on the legend, this is over time, where the last case in the red was found in 1st of August 1936 in Buffalo, Oklahoma. That was a distance of 525 miles over 148 days, and they determined that the average rate of dispersion was 35.4 miles per week. As John mentioned, this was typically what they used for their ecological studies, the Bishop trap. And they also used it as a control measure. They would bait them with lean beef or liver, and they'd add a little bit of water to keep it moist. The lower left photo is the results of one Bishop trap in Costa Rica, the beer bottles for scale, and this shows how many flies were in that one trap. Now, if you look to the right of that beer bottle, that's how many flies were screwworms out of that pile of flies. It's a very labor-intensive way of doing this. And Parrish in 1937 in Texas said that, found that typically one trap would have over 4,000 flies a day. So huge amount of work. And as John showed you, the huge mound that they had to go through. Again, this is our current screwworm trap. It is a vertical sticky trap developed by John and Renee Garcia. It's black because that's supposed to be more attractive to the flies. They did a study of looking at different colors of trap. Black seems to be the most effective. We use as John said, we use the Swarmlore 4. It's a chemical attractant that contains 10 volatile compounds in it. And that's what we use today and here in Panama. 1962, Hightower out of Kerrville, Texas, did a study where he was marking the flies. So for the first time in the 60s, we started to look at population dynamics. We could find the local distribution of screwworm flies, what habitat they were going to, and the distances that they moved. And this is a photo of the kind of habitat that they had there in semi-arid Texas. In this area, flies tend to orient themselves towards water. There was little fly activity when it was hot and dry, and there was an increase in activity after rains. Apparently, after 10 days, once it rained, 10 days, there would be high activity of screwworm in this area. And they also determined the recovery of released female flies was rarely exceed 0.5% of what they released. That's also what we have here in Panama as well. We release flies regularly to look at their fitness or to test different diet ingredients. And typically from the ground release, we get 0.5%. From the aerial release, we get 0.008%. So we even get less from collections from our aerial release. So this is just showing that the technique that Hightower used of using a fluorescent dye we still use now to test the fitness. We'll test, we change something in the diet, we'll test that to see if it behaves similar to the strains that we use now. And for the adults, the fluorescence is put on the pupae, and the adults get them as they emerge, excuse me. The first use of satellite data in the Screwworm program was in the mid-1970s. This was done by a NASA scientist, Charles Barnes. They proposed to model the areas favorable to screwworm using temperature and moisture from weather satellites. At that time, what was available was the NOAA Improved TIROS operational satellite, and they did the study for Mexico. It was not successful, and that's really because of the type of data that they had. It was very coarse. It had a very low spatial resolution, 1 kilometer to 4 kilometers, and also had not a very good spectral resolution. There was 2 visible channels and 1 thermal infrared channel. The photograph here is an example of the visual channel and the infrared channel for the eastern United States. I think you can depict Florida there easily, but it's very coarse resolution. As the eradication program progressed and eradicated from the U.S. into Mexico, so did the research progress to the south. Now the ecological studies are no longer focused in semi-arid habitats, but they're tropical habitats in the 1980s. There were several studies in Mexico and Belize. These were done by Bob Mangan, Don Thomas, and John Welch. These are photographs of them in the tropics. And the difference is, of course, is that in tropical environments, screwworm persists year-round. In Mexico and Belize, they use sentinel animals with artificial wounds to capture the adult flies. They typically use 2 locations in open or 2 locations in forest and compared those. They found the highest screwworm activity was in forest and ecotones in Belize and Mexico. This is an example of the sentinel animals that they used. They would typically they would make a wound, put 20 L1 larvae on the wound, let it sit for 4 to 5 days, clean it off, reinfest it with another 20 of eggs that they collected, raising eggs that they'd collected from the wounds. They would use that larvae for the second wounds that they would infest. They would infest the animal twice, and then they would rotate to another animal. So that was kind of what they did at that time. And in Costa Rica, as the studies moved to Costa Rica, Frank Parker, John Welch came up with a way of not having to use sentinel animals because it's difficult. You have to take care of the animals. It's also not that humane of a thing to do. So they did a study where they found that rotted liver baited feeding stations could be used to study adult screwworm activity instead of the sentinel animals. In this particular study, they had sentinel animals and they had the liver. They were able actually to mark and release many more flies, 195 versus 33 at the liver sites, and for the females and for the males, 44 versus 0. Typically, males do not go into the wounds, so it gives a little bit more information about the population as well. So from that time on, from Costa Rica on, they only use the rotted liver-baited feeding stations for screwworm studies. In Costa Rica, they also looked at how habitat influences the behavior of screwworm. They found that both edge and forest sites were equal. They're much more favorable than pasture sites. Remember, in Texas, they thought that the pasture sites was the most favorable area. But in the tropics, as we studied it, we find that that's different. Again, just to also point out that in this study, the forest sites were defined as 100 meters from the edge of pastures. This study is not designed to analyze all the habitats. It just used 6 observations, 6 trap sites. So, but they were really important observations. So the next stage for the research was John went to the Center for Safe Research, Melba Crawford's research lab, University of Texas at Austin in the early 1990s. That's actually where I met John. I was working with Eric Pianka on mapping fires in Australian deserts to study how that influences lizard population ecology. So that was when I first became involved in the program. At that time, to analyze satellite data, a workstation was required. You couldn't just use your computer. Now I just have a laptop, that's it. Also, Landsat 5 and Landsat 4 and 5 satellite data was what was available. You had to pay for it then. It was $5,000 a scene, or you could buy Spot, which is smaller, 60 by 60 kilometers, it's $2,500 a scene. So it was really expensive to do this work at that time in the early 90s. Now, luckily, USGS has all the Landsat data, historical and current, free download, so it's no longer an obstacle for anyone. So taking the observations of all the scientists previously who had done field studies, we went into Panama, had a spot image from 1993, sampled 116 vegetation sites to try to train the computer to map the vegetation sites for us. We were able to classify 9 different vegetation types in the Panama Canal area. And the reason for this, of course, is for eradication and control programs, you always have the question of where do you find the insects? How can we more efficiently and more effectively monitor this pest? We used, again, the decomposed liver bait traps. We sampled in 82 locations, 7 vegetation types from November 95 to October 96. We had a very large sample area of 55 by 40 kilometers. It was the largest scale. Spatially also had many locations and different vegetation types. So our results were that we had a much better understanding of the relationship of habitat and season on the geographical distribution of adult flies in the tropics. Edge forest was one of the vegetation types with the highest number of flies. In this study, we defined edge forest as being 200 meters from any opening. And so the nice thing about this, it's very easy to map on remote sensing data at many scales, aerial photography or satellite data. It can also be done visually using Google or simple classification programs. And it can also be taken outside the canal zone. So what we've done in the canal zone, I've also done for Cuba, and as well as for a Landsat image with Zapata County, Texas, mapping the areas of favorable screwworm habitat in that dry area. So it can be applied every place. Habitat mapping, however, is just one layer in the fly distribution model. We also need to have host density. So in the barrier zone, we have all of the farms mapped as well as a livestock census. So we were able to do a kriging model of looking at host density. We were able to combine that with favorable habitat to end up with a map showing The areas that are more favorable that you would have higher would be the reddest, is the highest density of animals there. Also, the favorable habitats are outlined there. And this is just an example of cases that we had in 2012, and they fall within this mapping technique. Current applications, really now we use Google Earth all the time. It's really easy to use. It's free. We can transfer data to all of our partners in Uruguay, Argentina, or wherever we're working. So it's a very, it's the way that we definitely start out with. This is just in a map here showing the Cora in the red. The yellow site is our field test site. We use this for determining where our traps go and our field sites. This is an example of the ground release chambers. So all of the studies that we do typically use the ground release chambers. They were fabricated here at the COPEG. They're improvised hard plastic boxes used in Panama for transporting agricultural goods. Each GRC holds 9.5 liters of pupae, which with 97% emergence is over 73,000 flies per ground release chamber. Each of the ground release chambers have 4 divisions in it, and each with 2.5 liters that are evenly distributed. We used this technique in Florida for the outbreak in 2016, 2017. We used regularly monitoring Google for doing the trap surveillance as well as the placement for the ground release chambers. For aerial dispersal, the technique that we use, we create a polygon in ArcGIS. I give the polygon, I make it into a KMZ Google file. I give it to our dispersal center. Ramses Rivera is our specialist there. He uses a program called Honeywell Sky Tracker software, and it draws on there the flight lines that would be needed for this area if we were to fly there. It lets you know that you can calculate the number of miles, number of linear miles, and from that we can determine the number of hours that it would take for the flight, so we would know how to bill that, and also the number of gallons of jet fuel. And these flight paths are 3.6 kilometers distance from each other. We used this in the barrier zone in 2008. The figure on the left was what we were flying. We were releasing 40 million sterile flies a week. Now we've trimmed down. We only release 15 million flies a week. We've trimmed down the barrier zone to reflect more of barrier zone instead of an eradication zone. We release 6,000 sterile flies per linear nautical mile in Colombia and 3,000 per linear nautical mile in Panama. We also use this technique for collecting a new strain. We had collection sites that were 10 kilometers apart. So and we also needed to find favorable habitats. So this was very helpful for that. And we used it also just recently last year in the study in Uruguay, looking at the effectiveness of rivers as natural barriers to the dispersal of screwworms. So we had dispersal chambers on each side of the river. We marked the flies. We had 6 traps on each side of the river. The lowest risk, this is also during the dry season, so this would be the time when the rivers were lower than they would be for the wet season. And the distance is between 2 kilometers and 4 kilometers in the depth. But you always need the field information. So we can define things and look at things using Google, but when you go to the field, there might be some barriers there. So one of the things that we're starting to do now is that we're using drones to help with our trap placement. So if we go to an area and it's changed, we can have the drone go up and look at the areas so that we can kind of direct where we want to put traps or a particular study. And thank you for your patience and your time and for joining us today. And I just wanted to thank also the people who contributed slides to this presentation. Okay, that's it. Hello?
At this time, ladies and gentlemen, if you'd like to ask a question, you can please press #2 to enter the question queue, or you may send a chat in the lower right-hand corner of your screen. Please send the message to all panelists. Once again, pressing #2 will allow you to enter the question queue where you can ask a verbal question. Otherwise, you may send a chat to all panelists in the lower left-hand side of your screen in the chat menu.
Okay, well, we've exceeded over 12 o'clock, and I don't see any questions in the written queue, and I don't believe we have any in the verbal queue.
No questions in the verbal queue at this time. Oh, we do have a question that did come up on chat. Let me send that to you.
The verbal question is, do Pamela or John know how far north screwworm has ever been reported? And how are the flies sterilized?
Okay, in relation to how far north, there are cases, humans got infested in New Jersey, and I believe there was a case in Canada at one time. Again, I don't know if I doubt that the flies made it up there on their own. They were probably taken up with an infested animal, but I believe there was a positive case in Canada. And then we currently sterilize using gamma radiation, cobalt-60, and we expose them to 50, is it 50 grays or 70 grays, Pamela?
55.
55 grays for about a minute and a half.
We have another written question. Are there plans to eradicate screwworm from the Caribbean?
There has been talk. Again, as long as screwworm is present anywhere, we are at risk of reinfestations and introductions. And so I'm hopeful that something gets started.
Okay, do we have any further questions?
We have one more chat question I just sent over.
Yeah.
And
John, would you send me further information on the Canada case if you can? That's from Joel Hutchison at canada.ca. I can send you that information, John, in an email.
Okay. And that will take, just for your information, that will take me a while because I lost everything on my computer, and so I'm starting from scratch. But I do remember from years ago that there was something, but I'll investigate and see what I can come up with. But that case was also, many years ago, back in the 50s or 60s. Now, with global warming, the situation is there's going to be more habitat that is favorable to the north in North America and also to the South and South America.
Okay, are there any further questions? Oh, we just have one come through. Has anyone looked at any ecological effects on areas where New World screwworm has been eradicated?
Not directly. Indirectly, we know that there was a study done in the Welder Wildlife Refuge in Texas when screwworm was endemic, and during that time period, they lost about 90% of the fawn crop of white-tailed deer due to screwworm infestation. And after screwworm was eradicated, we have more deer now. The tropics are a lot more complicated. It's a much more complicated environment, and we haven't really noted any differences there. About the only negative aspect that we say is there's a lot of cowboys that lost their jobs because they were out treating screwworm-infested wounds, and they no longer needed the cowboys. And then also in areas that have been eradicated, a lot of veterinarians have lost money due to having to treat screwworm infestations, and also the sale of insecticides to spray wounds.
And just to add to that, from studies that they did in Costa Rica, collecting pollen from the wild flies. Again, remember that there are very few in the wild. There's very few per mile in the wild, but also all the pollen that was collected were just generalists. They just typical plants. There was nothing in particular that they were specific to. Just to add to that.
Okay, we're almost finished with our time here. If you have any additional questions, please be sure to touch base with John or Pamela or myself. I'd like to thank everybody for attending today. We do have another webinar coming up on May 21st, and the discussion will be around euthanasia in an African swine fever outbreak. And with that, I will wish you all a great afternoon. Thanks for joining.
Thank you.
Thank you.
That concludes our conference. Thank you for using AT&T Event Conferencing Enhanced. You may now disconnect.